Hello. Today we are going to be studying the muscular system and the basic physiology of the muscular system. Okay, so to start with, each muscle fiber contains many myofibrils. Okay, you guys keeping up? I think before we even start with that, we should probably explain that each muscle is made up of heaps and heaps of muscle fibers or muscle cells. These cells are a multinucleic. I don't know what the correct term is. Polynucleic, maybe. This means that they have multiple nuclei, right? And there are 3 different types of muscle in our body. First, there is skeletal muscle. Skeletal muscle is the most common type of muscle. It's what you probably think of when you think of muscle. It's the muscles that are attached directly to our skeletons and that we have direct control over through our central nervous system. Alright, so skeletal muscle, we have direct control over it, okay? Think your biceps. And that's it. Okay, next is our cardiac muscles, and as the name suggests, those are just the muscles that make up our heart, right? Pretty simple. We don't have direct control over that. That's obviously completely operated and maintained by our peripheral nervous system, not our central nervous system. So the brain doesn't really have any part in your heartbeat. And then last we have smooth muscle. And that's just like that's all the other types of muscle that make up your organs, your organ systems, and kind of just also a lot of the time they line your organs, provide structure to your body, and yeah. My notes are much more in depth. I kind of sound like an idiot right now, but I'm just trying to go over the basics just so that I get it, I'm just going over my notes. Okay, so with skeletal muscle, such as biceps brachii, it's the basic. I'm not very good at this. Skeletal muscle, right? Each muscle contains many fibers, and they basically, on a microscopic level, they look like a bunch of little tubes running together. They make up a muscle. So each fiber basically contains many nuclei, even when they're just single cells sometimes, have more than one nuclei. And, okay, they're made up of L bands and A bands. Alright, so when you look at skeletal muscles, you'll notice that they are actually, the fibers at least, have dark and light stripes, so they're striated. And so the L bands, it's actually I bands, not L bands. I bands are light, and they are made up of thin filaments. And what does it say? Oh yeah, the Z disc is in the middle, is a midline interruption. The Z disc is a part of the I bands. Alright, then there are the A bands, and those are the dark parts, and they are predominantly thick filament. Okay, so one basic unit of a muscle cell is called a sarcomere. The prefix sarco will come up a lot in the muscular system. Probably means something like muscle or something. I don't know. Anyways, sarcomere is very similar in structure to a telomere, which, if you've done any biology, you'll know that a telomere very common structures within most cells in our body, and yeah, they work very similar actually, except sarco, muscle. Huh. Okay, so what does this say? Oh yes. Okay. So essentially, there are thick filaments and there are thin filaments. The thick filaments are known as myosin, and the thin filaments are known as actin, right? Okay. So myosin is composed mostly of the protein myosin, right? It contains actin binding sites and ATPase enzymes to split up ATP to obviously release energy, because to contract, your muscles need energy. Everything in your body that requires movement, or the movement of something usually requires energy, ATP. Myosin also have a lot of little bumps around them, and those are called myosin heads. And okay, on to actin, right? Actin is it's composed mostly of, what does it say? Contractive protein actin. That makes sense. Yeah, that makes sense. And okay, actin is anchored in the Z disc. So remember I said before that in the thin filaments, the Z disc is found within the thin filaments. Yeah. I'm just reading my notes again. I have very messy writing, so I've actually got to take some time to just decipher it. Hey. No, like I know this is just like dot points. These are just dot points. This is like kind of hard to explain because it's missing vital information. All the actual information on how it works is on the next pages. Okay, so here we're up to the part called stimulation and contraction of single skeletal muscle cells, right? So each muscle cell usually has 4 characteristics that can be measured and compared and contrasted. So we have irritability, contractibility, extensibility, and elasticity. So irritability essentially just means how easy it is to essentially like spur on the reaction of contraction, right? How easy it is to tell that muscle to contract. Then we have contractility, which is obviously it's how fast or like how strong it can contract to. And then extensibility, how far can it stretch from its original position, and then elasticity, how well can it return to that original position once it's no longer being contracted. Okay? So, we know that for skeletal muscles, motor neurons attach to the muscle, right? You know that. Everyone knows that. But they actually don't touch. No. There is a synapse there called the neuromuscular junction. Just like how there's a synapse in between all neurons, right? Called the synaptic cleft. There's one between motor neurons and the muscle called the neuromuscular junction, right? Easy. That's easy. Yeah. Okay, yeah. So, as we know as well, signals are transferred through neurons, or throughout the body really, by hormones, and when it's specifically a signal passed through the nervous system, they are called neurotransmitters, right? So the neurotransmitter that is responsible for letting a muscle know that it has to contract is known as acetylcholine. Acetylcholine. I don't know how to say it. I just know how to spell it. And yeah, so as the electrochemical signal flows to the motor neuron, flows through the motor neuron, and eventually reaches the neuromuscular junction, calcium ions, just calcium, I think, yeah, flows into the motor neuron at the end of the motor neuron, and it causes the little vesicles inside the ends of the motor neuron to release the neurotransmitter acetylcholine, and then that neurotransmitter diffuses across the neuromuscular junction to the muscle, specifically to the, I forgot what it was called. It was this weird word. See, that's why I have to reread these notes. What was it called? No worries. We actually don't even know. It's actually a mystery. Sarcolemma. Yeah, that's that. There's that prefix again. Sarco. I wonder what that means. Probably muscle, right? Okay, so yeah. The neurotransmitter diffuses across the neuromuscular junction until it hits the sarcolemma of the muscle, and then from there, the exact same thing happens when a electrochemical signal is sent through a nerve cell, a neuron, right? Where, imagine that it's a salty banana, right? Salty banana. Lots of potassium on the inside, lots of sodium on the outside, and once it reaches that action potential, once enough energy has been, once it reaches that action potential, the anyways, salty banana. During depolarization, sodium ion channels open, and lots of sodium flows into the cell, right? And that raises the charge. So initially the charge of the cell is very negative, and that's why it's called a polar. It's polar, considered polar, because the inside is very negative and the outside is very positive. So they're different. So it's polarized, right? Whereas then, when the sodium ions flow into the cell, the charge is increased, and it's called depolarization because the charge is now positive, and so is the outside of the cell. So now they're not different. They're not polar. Okay, and yeah. So once the action potential has been reached, let's read these notes word for word. Action potential causes the release of calcium ions into the sarcoplasm, and sarco. Oh my god, it's like the cytoplasm, but sarco. And bind regulatory proteins on thin film bind to regulatory proteins on thin filaments and expose myosin binding sites, allowing myosin heads on the thick filament to attach. So essentially, maybe I'll put some little things up there, but essentially, right, how it happens is there are those little buds, the myosin heads, myosin attachment sites, I think. This is the whole point of me doing this. It's for me to actually learn it, because I don't quite understand it yet. Anyways, so the calcium sorry the calcium ions, yeah, now I remember, they attach to a protein binding site, and that they act as enzymes, and what they do is they trigger an ATP to come and bind to the myosin head, and that ATP then releases a energy, and then turns into ADP, obviously, and inorganic phosphate, and after that it puts the myosin head in a cocked position, which then it's like a cocked position, and here's the thin filament, right? And you got to imagine, here's the myosin head, here's the thin filament, myosin head lies on the thick filament, the myosin, right? And basically then what it does is it pushes the actin forwards, and causes it happens on both sides as well. Usually there's a gap here where there's no actin, causes them to come together when they touch. That's why when you look at muscle cells, skeletal muscle cells, it goes dark, light in the middle, and then dark again, because there is no actin in that middle bit. However, when it contracts and these come together, that dark bit actually goes away. Maybe I've got the colors mixed up. Hey, yeah, maybe I've got the colors mixed up. I don't know. Anyways, a color goes away. That's all you gotta know. Yes. It makes sense in my head, but then trying to explain it, I sound like an idiot. Okay, okay. Let's read it step by step again. I found some more notes, yeah? Alright. Okay. So this is what happens, right? For a muscle to contract. Okay. So a nerve impulse reaches the axon terminal of the motor neuron, right? The motor neuron that's next to a skeletal muscle, okay? Calcium channels open, and calcium ions enter the axon terminal of the motor neuron. Calcium ion entry causes some synaptic vessels to release acetylcholine, right? And then acetylcholine diffuses across the synaptic cleft and attaches to the receptors on the sarcolemma of the muscle cell. That's basically what it's saying, right? I just was very convoluted about it. If enough acetylcholine is released, the sarcolemma becomes temporarily permeable and depolarization occurs, right? So essentially depolarization, as I was saying, sodium ions flow into the cell and causes the charge to go way up. Okay, so depolarization opens more sodium channels and the electrical impulse is transducted. Okay, then, bear with me, this is a very hard word. Acetylcholine esterane, yeah? Acetylcholine esterane breaks down acetylcholine into acetic acid and choline. Wow. Acetic acid and choline. Cool. And then the acetylcholine esterase ends. Oh, that is so messy. And muscle contraction. Yes. Makes sense. And then the cell returns to its resting state, right? Cool. What causes filaments to slide? Okay. So, okay, now let's go over the several different ways that ATP can be produced inside of the muscles, right? Because obviously our muscles need energy to contract. So there are three main ways that our body provides enough energy for our muscles, right? So one, probably the simplest way is direct phosphorylation of ADP by creatine phosphate, right? This is the fastest method because ADP is literally ATP, except it has one less phosphate group, right? Adeno-diphosphate versus adenotriphosphate. I probably butchered that. It's okay, right? Okay, then creatine phosphate supplies are exhausted in less than 15 seconds, right? So it doesn't really last that long. And on top of that, only one ATP molecule can be produced per creatine phosphate molecule, right? So it's not really efficient, or at least resourceful, you know, because as we'll see later, there are ways that produce much more ATP. And then we have aerobic respiration, right, which is a very long and complicated process. But essentially, all we'll go into right now is that it's oxidative phosphorylation, right? So it uses oxygen, and glucose as well. So it requires fuel you need to eat for aerobic respiration to be possible, and breathe. And okay, 32 ATPs are produced by this method, but it is a lot slower than direct phosphorylation through creatine phosphate. It's quite slow, but it is the most cost-effective one, essentially. And then we have anaerobic respiration and anaerobic respiration. So we have aerobic and anaerobic, and that just means respiration without air. So that's the process of breaking down glucose without oxygen. And glucose is broken down to pyruvic acid to produce 2 ATP, and then pyruvic acid is broken down to lactic acid. That's why if you've been working out for a long time or running, whatever, you start to feel the burn because it's literally acid in your muscles. How cool is that? I don't know. It's a cool little fact. And yeah, so that was my very long-winded and convoluted and not very clear attempt of going over my notes for the physiology of the skeletal muscle. So, yeah.